method used in modern biology. DNA barcoding uses a short genetic marker in an organism's DNA to identify its species. Think of it as a genetic barcode unique to each species. The technique offers several advantages. It allows researchers to identify species from tiny tissue samples. It works even when traditional morphological identification is challenging or impossible. The process is standardized and accessible to researchers worldwide. DNA barcoding uses standardized genetic regions that are specific enough to be unique to a species, but conserved enough to be easily identified. For animals, scientists typically use the mitochondrial gene cytochrome C oxidase 1, or COI. Plant barcoding often relies on regions like RBCL and MATK from the chloroplast genome. The DNA barcoding process involves 4 main steps. First, DNA is extracted from a tissue sample of the specimen. Next, the target barcode region is amplified using a technique called PCR. The amplified DNA is then sequenced to determine its nucleotide order. Finally, this sequence is compared to reference databases like the Barcode of Life Data System, or BOLD, to identify the species. DNA barcoding has numerous practical applications. It enables rapid biodiversity assessment, helping researchers catalog species in various ecosystems. It's a valuable tool for conservation efforts, allowing identification of endangered species and monitoring of populations. In the food industry, it helps detect mislabeled seafood and other products. The technique also aids in early detection of invasive species that can threaten native ecosystems. DNA barcoding has revolutionized how we identify and classify species, providing powerful tools for researchers across many disciplines. Genetic ecology integrates DNA barcoding with ecological research to understand ecosystem dynamics and biodiversity patterns. By using barcoding techniques, researchers can conduct comprehensive biodiversity surveys that reveal previously unknown species interactions and community structures. This approach has uncovered thousands of cryptic species, organisms that look identical but are genetically distinct. DNA barcoding has transformed our understanding of food webs by allowing scientists to analyze gut contents or environmental DNA to determine what species are eating. This allows researchers to track how energy flows through ecosystems with unprecedented accuracy. Genetic ecology enables monitoring of ecosystem health through techniques like metabarcoding, where bulk samples or environmental DNA are analyzed to detect multiple species simultaneously. This technique has proven invaluable for tracking changes in community composition over time, especially in response to environmental stressors like climate change or habitat fragmentation. Conservation biologists use genetic barcoding to identify cryptic species, organisms that look identical but are genetically distinct. Additionally, conservationists prioritize areas for protection based on genetic diversity patterns, which are mapped using barcoding techniques.